And welcome everyone to the inaugural webinar of the BC Living Labs Cattle and Forage Winter Webinar Series, running every Tuesday through and including February 27th. I'm Karen Raven, BC Cattlemen's Association Liaison to the BC Living Labs Cattle and Forage Joint Project, and I'm co-hosting with Wyatt Klopp, Acting Manager for the BC Forage Council, and Christine Cooper, Communications Specialist with the Investment Agriculture Foundation.
I have the pleasure of introducing our Living Lab. So the Cattle and Forage Living Lab, it's a partnership between BC Cattlemen, BC Forage Council, UNBC, TRU, and Agriculture Canada. Move on. So why are we looking at BMPs for cattle and forage? We want to just have this first and foremost. So winter feeding costs are the highest variable cost to cattle operation and subject to major fluctuation from climate change driven weather events such as drought, fire, flooding, things that we're very familiar with in BC these last few weeks and definitely the last few years. There's a lack of regionally specific information on how best to implement these BMPs for extended grazing in our geographically and climatically diverse province. And it adds risk to producers and hinders adoption. So we've outlined our study using two mother sites, what we call mother sites or core sites. We'll be hearing from them shortly. So the Devick Ranch down in Heffley Creek, Kamloops area, and the Barkay Ranch up in Prince George. We're also now hoping to implement daughter sites that'll be looking at 1 to 2 of these BMPs. Still working the kinks out here, but we have Tantalose Ranch out in Grassy Plains, Double C Ranch in likely Horsefly area, Double Q Farms down in the Lumbee area, and Pavan Ranch down in Grand Forks. So we're trying to get a bit of a breadth of implementation across the province. So some nice photos for you. So these are taken from our mother sites. We have the Barkay Ranch on the left, and then on the right, we have the Devick Ranch. And Paul Devick is here to talk a bit later about their research this past summer. So the BMPs, what we're interested in, so cover crops, we've done some replicated plots, fall seeding, corn grazing, and bale grazing. So we're really, this was just the first year of implementing these BMPs. So if there's anybody in the audience that has questions or suggestions for rooms for improvement, we're always looking to improve on these BMPs as well, or add that regional information. So as I mentioned, this was very much a collaborative project with the AIFC research team. So we'll be hearing from Dr. Shabtai Bittman and Dr. Kirsten Hannum. We also have researchers from TRU, Dr. Lachlan Fraser and Tanya Brewers, master's candidate. We have researchers from the University of Northern British Columbia, Dr. Michael Preston and Teresa Adesanya. And of course, we have our producers, Paul, Alex, and Stephen Devick from the Devick Ranch and John McAvoy from Barkay Ranch. In addition, we also have an advisory team of agricultural folks that help us with the agronomics. So we have Dr. Vern Barron, Jillian Bernard, and Darren Burgell. At this point, we'll now move on to our formal presentations. So we'll start off with a talk by Dr. Kirsten Hannum.
Okay, so thanks so much, Karen and Wyatt, for the invitation. I'm really delighted to join you for the first of many wonderful webinars that you'll be holding over the next, well, Paul Devick reminded me, for more than a month. So there's going to be some really interesting material that we get to see. I, along with my colleague Shabtai Bittman, have the pleasure of being the leads on the Agriculture and Agri-Food Canada side for the BC Living Lab. And we are really enjoying working with Wyatt and Serena before Wyatt and Karen and all the other wonderful farmers and collaborators on this project. So thanks so much for the invitation. Could I have the next slide, please, Wyatt? Okay, so I've been asked to cover 2 really quick topics. First of all, what is soil carbon? Where does it come from? And why do we care about it? And secondly, what do we mean when we talk about soil health? How do we measure it? How are we going to measure it in a living lab? And what can we do to protect or improve soil health? Next slide, please. So first, what is soil organic carbon? Where does it come from? Well, this is just a review for all of you from elementary school that plants do have this amazing ability to combine carbon dioxide from the atmosphere and water that they've taken up by their roots. And through the power of photosynthesis, they can produce organic carbon molecules, like the glucose molecule that you can see in that formula on the right hand of the slide. So thanks to photosynthesis, plants get organic carbon compounds that they can use to produce the leaves and seeds and roots and fruit that we all are grateful that they can produce. And as this plant material or this plant biomass dies, or as animals that eat that plants poop on the ground, then that organic carbon that the plant produced through photosynthesis is deposited into or on the soil surface. And most of this organic material will then decompose and be turned back into carbon dioxide and released into the atmosphere again. But a small portion of it is going to end up remaining for a little while in the soil as soil organic carbon. So ultimately, plants are the factories that produce soil organic carbon. Next slide, please. So why do we care about soil organic carbon? A lot of us have heard in the media these days that policymakers and researchers are interested in soil carbon for the fact that plants have this amazing ability to use carbon dioxide from the atmosphere and convert it into biomass. And because of this, we've been wondering, can we help increase the amount of plants that are growing and the amount of photosynthesis that they are doing, and then increase the amount of organic carbon that those plants have produced, increase the amount that goes into the soil, and just slow down its decomposition in the soil so that maybe we can start to draw down a bit of that excess carbon dioxide that is in the atmosphere. But that's not the only reason why we might be interested in soil organic carbon. Next slide, please. This is a map taken from a paper that was published just a few years ago, where the authors tried to estimate what the effect of management practices like agriculture has been on soil organic carbon over the last few hundred years. And when you look at the map, it's pretty intuitive to interpret. So the areas that are in red indicate regions where management practices have reduced the amount of soil organic carbon. Areas in yellow are where there's been a small reduction in soil organic carbon, but you can also see there's areas that are blue, and those areas are where our management practices have actually increased the amount of soil organic carbon. So it's amazing that we do have the ability to alter the concentration of soil organic carbon in our soils, either by accelerating its loss or promoting its accumulation. Next slide, please. And why might we even want to increase soil organic carbon? Well, it's magical stuff. If we had two soils that were exactly the same, except that one had higher concentrations of soil organic carbon, we would see that that soil organic carbon rich soil would have a lower bulk density. So that means there's less soil packed into a given volume. And because its bulk density is lower, it's more porous. And because it's more porous, it's more able to absorb water from irrigation or from rainfall. And that is really to the benefit of plants. It also is more able to hold on to the water that is absorbed by the soil, which means we lose less water to leaching. And because soil organic carbon is the food for bacteria and fungi in the soil, it's more biologically active. So it's being broken down constantly. It's got lots of living things in the soil. And we know that we like to see that in a soil, that it's a living thing. And as these organisms are breaking down that soil organic carbon, it's releasing nutrients, which can then be taken up by our crops. And as those nutrients are released, the soil does a better job when it has more soil organic carbon of holding on to those nutrients, rather than letting them leach out into the water, which is great for our soil fertility. And also at the same time, a soil organic carbon rich soil tends to have a better buffering capacity, which means it's less susceptible to acidification. And we know that as soil acidity increases, our nutrient availability tends to decline. So all of these are really positive effects that soil organic carbon can have on our soil. Next slide, please. So ultimately, soil organic carbon tends to be linked pretty tightly with our ideas about soil health. But what is soil health? If we were talking about this even just a few decades ago, we wouldn't really use the term soil health. We'd probably talk more about soil fertility or soil tilth. And these terms refer more to how available are nutrients in the soil and how easily managed is that soil. Is it easy to seed? Is it easy to drive a tractor over? But over time, we started to think maybe soil fertility and soil tilth, these terms don't completely capture what we value in our soil. So we started to use other terms. Next slide, please. So more recently, rather than using just terms like soil fertility, we've absorbed that term into soil quality, which covers not only the values we talked about when we spoke of soil fertility, but other things as well. So do our soils protect water quality, or do they tend to cause water contamination, say with nutrients? Does the soil protect habitat quality? Is it good for growing native species and crop species? Is that soil susceptible to erosion? Is the soil harboring high concentrations of pests and diseases? So soil quality captures more of the values that we have when we think of a nicely functioning soil. But even so, we started to realize that soil quality still isn't a large enough term to cover what we mean when we talk about the values that soil does for us, the services that it does for us. So that's when the term soil health started to be used more commonly. Next slide, please. And so when we talk about soil quality, soil health, we're talking not only about soil fertility and soil tilth, not only about soil quality, but also other services that a soil might provide. How good a job does it protect biodiversity? Does it support climate buffering? So is it absorbing more carbon dioxide than it's releasing? Is it suppressing nitrous oxide emissions? Is it doing a good job of helping protect our crops and our other plants when we have weather extremes like floods and droughts? And how, in general, does that soil help support our well-being right now and into the future? So ultimately, there is no one definition of soil health, and its meaning depends on who you ask, what they're worried about, and what they care about. Next slide, please. So this is a big question for us because as leads on the BC and the Peace Region Living Labs, we wanted to come up with some measures of soil health that would be meaningful to all the farmers that we're working with. And I can tell you, after talking to forage seed producers, cattle producers, dairy and vegetable and fruit and grape producers, everyone cares about soil health. A lot of people define it in different ways, but ultimately it is a value that we all hold really dear. Next slide, please. So the Soil Health Institute, which is based in the United States, has been pondering this question for about a decade. And they've recently done a really neat study where they collected samples from across North America, including several sites on the prairies and in Ontario. And they subjected these soils to a huge suite of common indicators of soil health. So I think they evaluated 30 common soil health measurements. And they wanted to find the minimum number of soil health measurements that would give you the most information. So the greatest value for the least amount of work. And they were looking for indicators of soil health that were responsive to different agricultural practices, that were really quick for a lab to analyze, or relatively quick, and that weren't redundant. So they were looking for a set of indicators that would provide unique information. And then they developed a set of protocols that they've made publicly available and that can be adapted for different labs depending on what equipment they have. Next slide, please. So this is the approach that we've decided to take, and several other living labs across the country are also taking, including the living lab that Vern Barron and Darren Brugel are working on in Alberta. So that's really exciting because it means measurements that we take will be meaningful to participants in the Alberta Living Lab and vice versa. So the three measurements that we are going to apply to samples collected from all living labs across BC, or living lab sites across BC, including the cattle forage sites, are measures of the concentration of soil organic carbon, which we know is a great indicator of many characteristics of soil health. Aggregate stability, so how well does the soil hold together when it's exposed to water? 24-hour rate of carbon dioxide production, which speaks to how biologically active is the soil, how much juice do the bacteria and fungi in that soil have to eat, and how busy are they in that soil. And Darren Brugel pointed out to me that he really likes these three indicators because they speak to chemical properties, physical properties, and biological properties of the soil, which hadn't occurred to me, but I thought it was an interesting point. So those are the three main indicators that we are measuring. We're also looking at whether we can do some analyses of soil moisture retention, because we know as soil organic carbon concentration changes, the soil should do better at holding on to water, which is really important when we face increasing drought frequency, for example. And then there are a whole host of other indicators of soil health that might be interesting to different commodities within the BC Living Lab, and they just depend on who's looking. So there may be capacity, depending on who is involved, to look at other indicators, but there's guaranteed to be the three that I mentioned at the top of this slide. Next slide, please. Okay, so what can we do to protect or improve soil health? There are a whole host of things, and I know that there are several management practices that the cattle forage sector are interested in looking at, or evaluating, or trialing and refining through this living lab. But I wanted to give an example that we've seen within Canada. So this figure is taken from an environmental indicators report that Agriculture Canada published, I guess, 7 years ago. And the figure that I've included here shows trends in summer fallow and no-till from 1981 to 2011. So essentially, these two practices have been strongly related to changes in soil health. By reducing the amount of time that the soil is left bare, or helping to keep it covered by growing plants as much as possible, and by reducing the physical disturbance of the soil by adopting no or reduced till through time, it looks like a lot of farmers in Western Canada have made some really interesting changes to their soil health. Next slide, please. So just by empowering farmers with knowledge and the equipment and tools to try and adopt these practices, there are estimates by Agriculture and Agri-Food Canada that suggest that through these practices, farmers have helped to maintain or even increase the concentration of soil organic carbon over this period. This is really a massive success story that is hailed internationally, and it really shows that just through group work and some knowledge in trying to find ways to increase the amount of time that plants are growing on the surface, and by protecting those soils wherever possible from physical disturbance, there have been some really strong trends in maintaining or improving soil health. Next slide, please. Okay, so this is my last slide. Just to conclude, why do we care about soil organic carbon? There are a whole host of reasons, but the main one for most of us is because soil organic carbon is strongly linked to soil health. And what do healthy soils do? It really depends on who is asking and what they care about. But ultimately, healthy soils function the way we want them to, long into the future. And that's it for me. Thanks.
I'll introduce our next speaker, Shabtai Bittman, Dr. Shabtai Bittman from Agriculture Canada. Lead the way, Shabtai.
Thanks very much. Karen asked me to talk about short-term and long-term research, and decided to do that with a focus on nitrogen, taking into account that Kirsten was going to talk about carbon, and carbon and nitrogen are both kind of the focus of the Living Lab in terms of climate mitigation. So there's several people that contribute to our work. Kirsten, of course, is one, and Derek Hunt, long-term colleague, and Gary and Jesse. I don't know if they're on the call or not. Okay, next slide. Yeah, so in terms of agricultural climate solutions and Living Lab, nitrogen is important. It's important because it directly relates to climate mitigation and also sustainability of cropping system or farming systems, and obviously profitability as well. So next slide. So this is intended to show how much nitrogen is involved with the beef cattle sector, and we did this calculation a number of years ago. It wouldn't be too different now. And basically it amounts to 634,000 tons of nitrogen per year that's excreted by beef cattle. So how much is that? That's almost exactly a quarter of all the nitrogen fertilizer that's used in Canada. And of course, Canada is a major crop producing country, and fertilizer's a big story here. So this is a sizable amount. Now, next slide, please. So what happens when you introduce nitrogen into any kind of a system, a field, a farm? You have inputs and you have outputs. And there's a kind of a balance or an imbalance between the inputs and the outputs. And if you have inputs exceeding outputs, then you're going to have losses in the system. And the losses with nitrogen are usually ammonia, N2 gas, which is the atmospheric nitrogen, nitrous oxide, and you can also have leaching and nitrate and runoff of organic and other nitrogen focuses. So if your imbalance is there and you try to mitigate one of those loss pathways, you will probably get pollution swapping. So that nitrogen will escape some other way. So in order to achieve efficiency, you have to both block the loss pathways and balance inputs and the outputs. So next slide, please. So this kind of shows a leaky pipe in reverse in a way. It's an alfalfa stand. The picture is taken in I think it was in Minnesota. It's in our Who Forages book. So what they did was they injected smoke into a tile drain system, and where the smoke is escaping are the macropores, and those are the macropores either by wormholes or cracks in the clay or things like that. And it shows the rapid pathways for loss of nitrate into the aquatic system, if you will, or lower down into the soil. So it's kind of a neat picture. So next. So in terms of the cattle industry, so the excretion of nitrogen, that 634 that 1,000 tons I mentioned, so about half of that is excreted on pasture, and about half of it is excreted in confinement, in various kinds of confinement systems, feedlots, so dry lots, and so on. And of the nitrogen that's excreted on pasture, very little of it is theoretically lost. Only a small amount, maybe 5 to 10%, as ammonia. Quite different for nitrogen that's excreted in confinement. And when you look at all the different loss pathways, I won't go through them all, but only about a half is retained and about half is lost. And most of that loss is as ammonia. And if you look at the overall cattle industry, 27% of all the nitrogen in that system from manure is lost as ammonia. So next slide. So what happens to the loss? Well, this represents the entire ecosystem, if you like. It's called the nitrogen cascade. And in the The box in the kind of center middle, that's in green, that's agriculture. And then the nitrogen that's lost is the nitrogen that's lost goes up either into the atmosphere, into the water system. So what I wanted to focus on is the nitrous oxide loss pathway. It's in the yellow box that's blue, that's got a blue box around it. And that shows the direct loss of nitrous oxide, usually from manure and or fertilizer on farms. But there's a secondary loss pathway that can't be ignored, and that is the ammonia loss pathway. And so if you recall, there was a lot of and that gets converted into nitrous oxide as well. So that's called secondary nitrous oxide. There's also some there's also some of the nitrate that's lost by leaching. There's not probably that much in the cattle industry. A lot of it is on dry land. But nevertheless, that which is leached also will wind up being converted into nitrous oxide. So the combination of secondary and primary nitrous oxide are what the industry has to contend with. Okay, next. So first, let's talk about the short-term nitrogen measurements, because that was one of the goals of this talk. So nitrous oxide is something that needs to be measured short-term, and the next slide will show you why that is. This is 4 years of sampling of nitrous oxide at Agassiz, and it kind of looks the same wherever you go, kind of. But what it shows is how episodic the emissions are. You have very, very low levels of emissions when under typical conditions, but as soon as they're perturbed, either through application of nitrogen, soil disturbance, and freeze thaw, that's where those blue circles are. They represent freeze thaw cycles. Then you get spikes of emissions. In order to be able to track that, you need to take many measurements, and you need to a little bit anticipate when the spikes are going to be. And so this involves year-round measurements. It involves frequent measurements, and it's why in living labs we can't go to all the farms and do nitrous oxide measurements and get comprehensive values. So next slide, please. So this is how they're measured. This is how we measured it, typically manually with these vented chambers, and you lay them out, you put the covers on, you take gas samples, several of them, 4 of them, and in sequence to get the rate of nitrous oxide emission. And you have to go out there repeatedly to get those measurements. So next slide, please. So in order to contend with the variability over time, automated chambers are sometimes used. We don't have them. They do present their own challenges, but they're great in the sense that they'll get you a measurement every hour, every 2 hours. And this is a setup actually in China that I visited. But that takes care of the temporal variation, but there's also spatial variation. And with cattle, it's particularly significant because of urine patches. And urine patches would have something like 500 kilograms of nitrogen per hectare in a patch versus next to nothing adjacent to the urine patches. So it depends where you locate the chambers, you might catch one or not. So to accommodate that, there's a different system that's used. It's on the next slide. And another source of variation is the aggregation of the congregation of the animals, the aggregation of the animals. So they're not uniformly distributed across the pasture. This is survey data we got from actually surveying hundreds of farmers across Canada, and there was quite a reasonable agreement that they figured about 24% of the time the cattle on pasture are congregated around either watering holes or shady spots under trees and things like that. So that shows another reason why it's important, if one attempts to measure nitrous oxide, you not only have to look at the variability between urine patches, but where those urine patches occur in the field as a whole. Okay, so next slide shows you how this is sometimes done, which is a micrometeorological method. And this one is called eddy covariance method, and also a picture from China as it happens to have that handy. And there's only a few setups like this in Canada. There are University of Manitoba, Guelph, I think Lethbridge has one. as probably a couple of others. They're very expensive, and they cover large areas, and so they allow you to measure emissions off large areas, which is very good for pastures. But you normally wouldn't have more than 1 or 2 or 3 of these systems. The Living Lab in Manitoba has one such a system under Aaron Glenn. Okay, next. Talking about aggregation and congregation of animals, of course, this Living Lab is very concerned with winter grazing, and this is bale grazing, as you can see. And there's been an enormous increase in bale grazing over the years, and you can see the data comparing 2006 and 2012. These are surveys that we did across Canada on hundreds of farms, and you can see that it's very popular on the prairies. But it's also gaining some acceptance in other parts of the country where there's more snow. Okay, next, please. And as I mentioned before, it's year-round. It seems like we're a little bit cut off on the slide. I don't know if you can see it all, but what it shows is the emissions happening. This is in Quebec in the wintertime. It's cold and it's frozen, and you can see the red line in the top graph is the salt temperature and sub-zero. And if you can see any of the spikes there, the bottom graph, those are the nitrous oxide spikes. And you can see that there's a lot of spikes coinciding with frozen soil. And that's particularly true if the soil is fine textured. Also, you'll see that where the temperature of the soil goes up above zero on the right-hand side, there's a major spike. And that's a freeze-thaw thing. And it's very big item, especially on the prairies. It was discovered in Canada, and it's now recognized worldwide where there is frozen soils, of course. Okay, next. So in order to accommodate and to calculate how much emissions there are in Canada, there are things called emission factors. The next slide will show you the Canadian emission factors. They're default factors that IPCC allows you to use, but Canada has developed country-specific emission factors. These are the ones that I think are currently being used, and won't go into them too much except to show that, for example, in the middle in that dark blue shaded area, fine soils is 3 and coarse soils are about 0.6. So a big difference in soil effect, organic versus synthetic nitrogen. And you can see in BC at the bottom, there's the dry areas and the wet areas for emission factors. So there's all kinds of factors that will affect, and whether the nitrogen comes from nitrogen fixation or biological nitrogen fixation or fertilizer, that makes a difference too. Okay, next, please. So long-term nitrogen measurements, we're going to talk a little bit about that. Next slide. So the first thing I just wanted to show by way of passing, because I had this data here from work we done in Saskatchewan years ago, and what it shows is that there's a balance on pastures between the coarse grasses. Typically, those are the ones that are seeded, and those are the ones that yield better. In this case, it's smooth brome, and then the fine grasses are volunteer grass, and this was Kentucky bluegrass. I think that's in BC as well. And so what you can see is that there's about a 1-to-1 ratio when there's no fertilizer added. If you add straight nitrogen at 90 kilograms per hectare, then you get a shift to the fine grasses. If you add phosphorus, I don't know if you can see this at the bottom, it's cut off in my computer, but if it's, so if you add the phosphorus, then you improve the ratio. And at the very bottom line, if you can see it, there's phosphorus and sulfur. And phosphorus and sulfur, so the ratio of coarse to fine grasses goes from 0.6 to 1.7 to 3.5 or something like that. And that's the importance of balancing nutrients. And part of the reason I mention that is that parts of BC and parts of the cattle producing areas are low in phosphorus, and they're also low in sulfur. And sulfur deficiency, unless you test, is hard to discern because it looks exactly like nitrogen deficiency. So something to keep in mind. With less deposition of sulfur from the atmosphere, when car exhaust has been limited and factory exhaust, there's been less deposition of sulfur. So sulfur deficiency has become actually a bigger problem. Okay, next slide. So this actually shows you what happens in long-term trial. We've kept it actually quite a bit longer. I had this slide handy. But what it shows is that the N recovered by the crop for equivalent amounts of nitrogen applied with this manure fertilizer, it's higher with fertilizer than it is with manure. But if you look at what's retained in the soil, it's higher with manure by quite a bit than what's what's in the soil, and so what's from fertilizer, and that's really important. And this shows nitrogen numbers, but I could equally have shown you the carbon numbers that follow the same trend. So manure definitely contributes to carbon sequestration in our system here, and also nitrogen sequestration. And what happens to the rest, we did not measure per se, but we know that there's very little volatilization of ammonia from the fertilizers we use. There is from some urea, high volatilization. We use ammonium nitrate, it's very low. So it's pretty well all leaching or denitrification, probably all leaching, and in our high rainfall area, whereas most of the losses from the manure is actually volatilization. Okay, next to the end. Nitrogen is released, and it's released quickly. And so it's very important to try to capture that, because otherwise it can be lost by leaching, by runoff, and also by nitrous oxide emissions. And this shows the peaks. All of that nitrogen that's released there is from stored nitrogen that was in the soil in that grass from manure. Okay, next. So now we're getting right to the end here. We're going back to our leaky pipe and looking at the farm budgets, because that's how we want to pursue nitrogen and the nitrogen efficiency and the nitrous oxide emissions by inference by doing nitrogen budgets. So the next and kind of the last slide shows what these budgets look like for cattle industry. Also, there's items there that would relate more to other types of farms. But you look in the field inputs, and you can do this for field, you could do that for farm, and I'm hoping we'll do both. So field inputs are the cattle that's starting, fertilizer that's put in, manure if it's imported, and fixation, if there's other kinds of inputs that are added in. And the outputs for cattle, it's mainly the cattle that's brought out, but there may be other types of it's maybe some of the if there's surplus hay, that may be harvested and removed. And so when you do the nitrogen content of what's coming in and what's going out, then you can do a balance. And if you know what that balance is, or if you know, in other words, what the surplus is, from that you can infer the nitrous oxide emissions. It's been shown in the States to do that, and I think it's been effectively implemented there, and I think is what we want to achieve as well here. So that's sort of my last slide, and this is how I'm kind of circling back to what it is we're trying to do with Living Lab and with these measurements that we talk about taking.
We're going to start off with Paul Devick Ranch, who, as Wyatt noted earlier, has one of the mother sites out in Heffley Creek near Kamloops, BC. And we've asked him if he would share his on-farm research experience, and looking at, Paul, if you would describe your experience with on-farm research and the practices you tested. I know you tested a couple, and there's some great pictures of those, and also what you found worked really well, and what you would or will do differently from next year, both from, you know, the practical field standpoint, as well as in the overall communications with ranchers and researchers.
Oh, can you hear me?
Sure
I'm
can.
not on mute. Okay, perfect. Okay, thanks for that, Karen, that introduction. And we at Devick's Ranch here, there's myself and the next generation, Alex and Stephen, are most involved with it. We took on the project thinking that, okay, well, let's see what we can do for carbon sequestering and nitrogen usage and all these kind of things, which is a little bit overwhelming to begin with, but I think I'm starting to get to understand how the whole thing works. But anyway, so what we did is we have a new field that we just developed. About a quarter of it had some forest stand, a timber stand on it. So we cleared that and squared up the field and carried on with the project, fenced it, and etc. So on the one half of the field, we planted corn at a rate of 32,000 seeds per acre, and added 530 pounds of slow release nitrogen fertilizer on the corn, which, I mean, and we also added 10 to 12 tons of compost, which is made up of our feedlot manure and compost from our meat plant, which we have at the same time. So there's composted waste from the meat plant is mixed in there and done applied as well. One thing that really surprised me mostly, I mean, I'm kind of old generation, but really surprised me at the success of the corn crop that grew at our high elevation, which is about 3,000 feet in elevation, maybe a wee bit less, but not much. Years ago, we would never have got corn to grow there, and now with all the plant breeding technology and stuff, they've got these short maturing corns, and the corn just grew crazy. We had corn 9 feet high in that field, and for whatever reason, I don't know what we did right or whatever, maybe it's normal, but we were very happy with the crop that came up. Oddly enough, where the tree stand was, production was less, and we'll be looking at that quite a bit more through next year. I know the scientists and the people working with us mapped all that out through Google Earth and could see where the trees were and where the trees weren't. So that'll be very interesting. A lot of this stuff is just in works right now. This is our first year. So that then on the other half of the field, we planted a cover crop called Forage Pro, which has rye, millet, vetch, clover, radishes, turnips, brassica, kale, and beets in it. And so it's a real mix of varieties there. And then we planted 110 pounds of triticale with that. And in hindsight, we put way too much triticale in there, and I think it choked out some of the germination of the Forage Pro species. And so next year we'll reduce the amount of seed, and maybe even switch to barley. Our research team is meeting at the end of February to plan all this stuff out, and so that's will all be worked out then. So then moving on to the grazing, we grazed the Forage Pro crop first, and right after frost, and the cattle went crazy in there. They chewed everything down right to the soil, and a lot of the turnips or radishes. Anyhow, a lot of that was left in the soil for soil improvement, and so that worked quite well. And the cattle grazed all the top growth off, and it'd be interesting to see how the soil tests come out after that, as far as soil organic matter, and I guess all this carbon stuff, which I haven't quite got that all figured out yet, but it's coming. So anyway, and then right after that, we moved into the corn, and we divided that corn field into 4 plots, 5 plots, 4 or 5 plots anyway. They were about, the bigger ones were 5 acres, the smaller ones were 2 or 3 acres. And, excuse me, and moved the cattle from plot to plot, and probably fed them 20% of their diet supplement with hay, just because our cattle have never seen corn and didn't know how to graze it, and so that was very interesting to watch the cattle. But they, as soon as you opened them into a new block, they were in there like crazy, and really loved the chewing on the corn cobs. But the challenge we had there, we could have improved our fencing. We had a few wrecks with the fencing, and we're on it right away, so it wasn't serious, but that will definitely have to improve that for next year. But getting back to soil carbon and nitrogen, I think again, it's hard for me to comment because we don't have any of the results yet, and we'll know a lot more after our research team at the end of February. But I think it would be sure nice to reduce that 530 pounds of nitrogen fertilizer somehow, and we will be again applying some compost fertilizer or amendment, I guess. That's available for us as well, as it will be every year. And so the organic and the nitrogen holding capacity hopefully will improve. And I think as far as the carbon sequestering, we had very little exposed, and we did all our tilling was limited tillage. And so, I think, yeah, it's all very interesting. So other than that, if there's any questions, we were happy with the project. It would be nice to know, the results from our heat units that it took to grow that crop. Maybe we were just lucky this year because it was such a hot summer, but we put a lot of water on the field too. So I think we did 4 passes with our big gun at putting about 1.5 inches of water on at each pass. And you got to remember, we're at 3,000 feet elevation, so temperatures are not as warm as they are down in the valley. So I think that's it.
Great.
Wyatt or Karen, who's ever hosting.
Sure, yeah, really appreciate that. We do have one question. We'll just save it to the end. It was and another one from another participant. So we will just then I'd like to thank so much, Paul. I think that really gave a good idea of sort of the good and the bad of what you experienced. And I'll turn it over next to Tanya Brewers, researcher with Thompson Rivers University. And she worked with the Devik Ranch. And then we just posed a few questions to each of the researchers for, there's might be folks here on the call that are thinking about getting involved in on-farm research. And just if you could let us know some of the activities and equipment and some of the things researchers need for successful on-farm collaboration. So take it away,
Okay.
Tanya.
All right. So I'm Tanya from TRU, and I did a lot of work with Paul and Alex, and it was a very valuable experience. I would just like to say right away, it was a fantastic experience. And I think for the ranchers out there, I would like to say that our input wasn't a lot of equipment. We were using some soil augers, some bulk density testing. We were doing some veg surveys. So I would suggest that we weren't like an invasive presence on the ranch. So I would think it was fairly low impact. Other than that, I would suggest that some of the really beneficial aspects of this was the communication and the relationships. When we first started, I didn't know anybody, so we really worked to get to know each other, and that was helpful because before we have to get out and do some of our sampling, we do need some notice. It's important that we have some time to get our team together, and once that process was occurring, there was a lot of communication, and from this point forward, I do a lot of talking with Alex. So that's been fantastic. Other than that, some of the successes, I personally was very happy to see all that corn growing. Other than some glitches with the electric fencing, it seemed to be quite a success. So we did quite a bit of soil testing and veg sampling in the fall. From that kind of information, I'm going to be able to give the ranchers what their yields were and some of the soil chemistry and nutrient results. We're going to test again in the spring, and then we can sort of look at things too, like in addition to the carbon, we'll be looking at the nitrogen, like Shabtai was talking about. We'll also be able to tell at that point too what the intake was from the cattle. So that will give the rancher some idea of if there was success in the program. Other than that, I would just like to say overall it was a valuable experience from a researcher standpoint. And so I'm hoping that the ranchers feel the same way because we've really enjoyed it.
Great. Thank you so much, Tanya. That's really appreciated. So next we'll ask Dr. Michael Preston and Dr. Teresa Adesanya from UNBC, University of Northern BC, and they worked with the Barkay Ranch up in Prince George. So turn it over to Michael.
Yeah, thank you very much. So yeah, I'd just really like to echo what Tanya was saying there. One of the greatest things that we found working with Bouquet Ranch is actually getting input from the rancher themselves. You know, we're researchers, we're not ranchers. And so a lot of the ideas has really worked in collaboration. So I do see this as a big partnership between John McCoy at Bouquet and ourselves, because we couldn't have done it without him. And one of the things that we really like about the Bouquet Ranch is we're able to get replication of our sites on the plot that he's set aside for us. And that's really important from a scientific point of view, because as you know, if you're considering this kind of trial, 1 year or one particular part of your ranch might respond differently. So it's important that we kind of replicate it on the landscape so that we can see that these BMPs that we're employing actually have good and consistent findings. And then I'd just say, like, from an equipment point of view, it's we've really relied on John and Bouquet Ranch and their expertise and their ability to apply these BMPs. We obviously bring our own equipment to do soil sampling. And likewise, it's that open partnership between just letting us know what's happening and what can happen, say with the cows, with the cattle on the treatments, and when they need to be moved, and then we can respond to that fairly quickly. The advantage of having what we're terming these daughter sites versus these mother sites where the most of the experiment is going on is that can we actually see similar results on other farms, and can we relate that back to what we're finding, which is going to be really important for kind of making recommendations going forward.
Great, thanks very much. And then I'll turn it over to Dr. Teresa Adesanya, also from UNBC, just to close out our final 2 minutes.
Thank you, Karen. Yeah, so I'll also echo what Michael said. It was a really insightful experience, because we didn't have experience about beef cattle operation, but John was really helpful, and it helped us frame our research question very well. Also for the activities and equipment involved, aside from the equipment, clear communication is really key, because we would need to go sampling prior to harvesting, and John was really good with that. So that was very, very helpful. But overall, it's been a very rewarding experience, and the multi-year part of the project is really beneficial, because when we have our downs, we have another year to retest, or we can go back and re-strategize and do stuff better. So that's everything. Thank you.
Super, thanks so much. Really appreciate the insights from everyone, and hopefully that was helpful to the audience as well. And we are very close to the end here, but we do have 2 questions. And the first one, I think that will go to Dr. Shabtai Bittman. Can the nitrogen budget be improved by ensuring that as many factors as possible that are limiting forage yield are addressed?
Yeah, am I on? Yeah, you can hear me, right?
Yes.
Yeah, okay. Yes, absolutely. There are limiting factors, and I did make reference to phosphorus and sulfur, for example. So absolutely, you need to look at the whole picture in terms of trying to maximize that production to make the most out of the inputs. But if you can't improve your inputs, you have to reduce it, or you can't improve your outputs. Well, then you should reduce your inputs. So you still need to achieve a balance. So, for example, you may have a dry year. And so, your inputs may exceed your outputs. You hope you don't lose them. They may carry over to the next year. Anyway, there's all these considerations, but absolutely, it's not all just about nitrogen by any means. It's just that we're focusing on nitrogen because it's the most singly important input from a soil's perspective. And because it has these direct connections to greenhouse gas emissions. And I didn't mention, I think everybody knows that nitrous oxide is a dog out there. It's 200, 300 times worse on a 100-year analysis compared to carbon dioxide. There's not as much of it, but it's something that is, you know, is very agriculturally focused. So that's why we're kind of talking so much about nitrogen. But no, nitrogen without phosphorus, sulfur, and all the other potassium in many cases. So it all needs to be taken into account.
Super, thanks so much. And we just have one final question, and this will go to Paul. And it was the full name of the cover crop mix.
The cover crop is called Forage Pro. And it's you know, it comes all pre-mixed with all those things in it. We added some annual ryegrass is all we added to it. So but it's called Forage Pro, and yeah, got it from Purity in Kamloops.
Great, thanks.
Once
so
again,
much.
it's got rye, millet, vetch, clover, radishes, turnips, brassica, kale, and beets in it at different percentages. So yeah, it's a real mix. And you know, I like the fact that half of the turnip, or was it turnip or radishes? I forget which one. One of you can answer that, but half of that plant was above the ground and half of it was below the ground, and the cattle just chewed off the upper portion, and the bottom portion remained in the ground because of the frost, and that will all go into organic matter for building our soil and improving it along with the compost.
Great. What a wonderful way to end the webinar. Thank you so much, Paul. And thank you to all our presenters and also to everyone in the audience who joined us. And of course, we will put in a plug for next week's session, which is on bale grazing with Darren Brugel and Alberta rancher Doug Skeels. And we'll also get a chance to hear from BC rancher Kim Hardy as to why she's looking into testing that process. So it should be another interesting session, and we do hope you'll join us. And thanks again, everyone.